Just thought I'd give you a little snapshot. This is for those of you who don't know it, this is what was the Scottish Crop Research Institute and now the James Hutton, which is situated by the Tay near Dundee in the east coast of Scotland. And that's it on one of the very few nice days, probably, for aerial photos. So I'm going to try and give you an overview of what I do, but also in the context of what else is going on in the Institute. I'm not going to go in depth into, hopefully I'll give you a wide range of things, snapshots into some of the work that we're doing. And so you might have questions, feel free to ask at the end. And also I'll try and reference papers that you might, if you want, you can go and read in depth. And yeah, feel free to ask at the end if I'm too confusing. So I wanted to say at our institute we have a large plant pathology department and we're all really beginning to look at effector proteins, which I'll say more about in more detail. But there are people working on, we're interested in crops, plants, and there are people interested in bacteria, viruses, aphids, nematodes, and this thing here at our institute. And this is oomycetes, the Phytophthora infestans. So I'll try and describe a wee bit more about that, because until I did my PhD, I hadn't really heard of these organisms myself. So I'll tell you a wee bit about them and why they're a bit different. So the name of the organism that I'm going to talk about is late blight, is the name of the disease, and it's Phytophthora infestans. And literally it means plant destroyer. And it does exactly what it says on the tin. This is a patch of potatoes, and you begin to see an infection in the middle. And within a week, the whole potato patch is gone. So it's a really big problem in agriculture, and they're really struggling to control it. They can try and prevent it, though, with sprays. But a lot of the sprays that the farmers have been relying on are being withdrawn under new EU licensing. So we're really getting to a point where we're losing a lot of the most bioactive chemicals. And we're at a situation where we're struggling to find or have durable resistances in the potato plant. There are some resistance genes, however, and our institute does have a breeding programme. But the problem is that Phytophthora is not just one disease that stays constant. It's the same as flu or many other animal or human pathogens. It's constantly evolving in the field, and it changes depending on what the farmers are growing in the field. It takes about 25 years to breed, sometimes it can take up to 25 years to breed a potato. So if you're breeding for a disease that's here now, and it takes you 25 years to do it, the disease will have changed dramatically within that 25 years. So this is the problem that we have. And here is some data from some colleagues who basically they genotype all the isolates of this Phytophthora organism from fields around the UK, and then they try and organise them and how related they are into groups. And if I draw your attention to, say, this blue bar, so before 2005, this blue isolate, we call it blue 13, didn't exist, and it came in from Netherlands, but we blame the Netherlands for a lot of our isolates that come in. And within 3 years, pretty much it's dominating. 80% of all the isolates that we were getting back from the farmers were this one isolate. And this isolate breaks a lot of the resistances that we had bred at our institute and elsewhere. So it's a big problem, and I might refer to this one later, so you have to remember. So we call this one blue 13. There's another one we call pink 6 as well, which is this pink bar, and it's also increasing at certain times of the year and going down. So you can see them coming and going, and it depends on the weather, it depends on the varieties the farmers are growing. So it's fluctuating, it's fluid, and it's changing all the time. So there's no point having just one resistance gene in the field. It's not going to last, and when the thing you're trying to protect against is changing this fast. So here's one of our flagship cultivars called Sterling, and it has something called field resistance. And this blue 13 isolate I was talking about overcomes it, so it's wiped out our breeding material on this in one fell swoop. So we're having to rethink what we're doing. But you can see it holds up against some of the other isolates that we have. So I've stolen this quote from a colleague from the Netherlands who used it at a conference, but I quite liked it. If you know the enemy and you know yourself, you need not fear the result of 100 battles. So the point of using this is there's no point trying to breed resistance when we don't know what it is that the resistance genes are recognising and how stable they are in those populations I was talking about. We need to understand better the pathogen that we're trying to fight to know how well our resistances will last. And that's going to be the basically the principle of my talk. So my outline is going to be how do we understand the enemy? And I'll talk a wee bit about some of the projects we have on that. Discovering what it is the Phytophthora wants to manipulate within its host, and then how we're using that information and genomic information to try and identify host resistance. So, understanding the enemy. So I said already that Phytophthora is this organism, and you can see it's fluffy, it grows fluffy colonies, and it has mycelia, and that's why for a long time people thought they were fungus. But actually, well, I'll say it was introduced a long time after actually potato. So potato came to Europe around 500 years ago from Peru by the Spanish, and UK and Ireland were some of the first people to really start growing it, especially in Ireland. Sir Walter Raleigh took a lot of potatoes and grew them on his own estate, and that's partly why Ireland was one of the first to really grow it a lot and start using it and eating it a lot. And then Phytophthora infestans came into Belgium from Mexico, probably with the slave trading and all sorts of things, but that's the circle. And it came in, and within the minute it got there, basically, it was causing famines. Hardest hit was Ireland, so you guys will probably know this memorial that you see along the river. It's really nice, quite a haunting statue, it has to be said. And that's because this disease is one of the most historically significant and did have one of the highest impacts on human population for a plant pathogen. And basically, places like our climate, Scotland, Ireland, nice, cool, 18-degree summers and wet, damp are paradise if you're Phytophthora. So that's why we have such a problem with it. So here's a tree of life, and this is just to show how far apart from fungus it really is. So here we have fungus, animals are down here as well. Here's plants, and it's way over here. And it's related to brown algae, so it's come out the sea. So it looks like a fungus, it grows like a fungus, but actually its cell walls are not chitin, or it's only 1% chitin. Fungus walls are chitin. This is cellulose, which also makes it quite similar to its host, which makes it difficult to fight. It reproduces both sexually and asexually, but I'm not going to get into the life cycle in too much detail. And it's not just pathogens of plants, there's also pathogens of algae, the fish, salmon, aquaculture having a big problem with it. And it can get into burns, and there's a lot of horrible ones that you don't want to look at. But the group, it's also, what I'm going to talk about is how it's devastatingly a dicot pathogen, and pretty much every crop, dicot crop's got a Phytophthora that infects it. And if anyone in here is from forestry today, we also have a big problem with it causing diseases in forests. And again, this is going to be harder to treat and spray, and how you prevent spread in natural environments, not just in agricultural environments. So there's a lot of reasons to study this organism. And one of the biggest tools that we have to help us was the genome sequencing and the drop in cost of genome sequencing. So here we have the introduction of next-gen sequencing techniques, and we published this Phytophthora infestans genome sequence in 2009, just as the cost started dropping significantly. And the reason for that is because the Phytophthora genome is 240 megabases. This is the bean one and the tree pathogen. You can see that this Phytophthora potato pathogen is a significantly bigger genome. But the other interesting thing is the number of genes isn't significantly bigger. And basically what's happening inside Phytophthora that we found when we were sequencing it is that it's a lot of repetitive DNA there. 74% of the genome is just transposable elements and repeating. So this gives Phytophthora a lot of potential to move its genome around, a lot of fluidity. It can pick up DNA, it can lose it, and that's partly why we have so many genotypes and it's so fluid. And just to put it in perspective, the model plant is Arabidopsis. Its genome's 135, so this beast that we have on potatoes got twice the size of genome as the model plant. So it is a big genome, and that's why it costs so much to sequence, and it's why we didn't sequence it until the price came down. Sorry, this is quite a full slide, but basically this is one of the figures from the Nature paper. And here's just what it is plotting the distance, 5' end, 3' distance of the genes to its nearest neighbour. And basically most of the genes, the housekeeping genes in Phytophthora, are very close by to another gene of interest. But this group of effectors that we're interested in, and they're called RXLR effectors, and that's because the RXLR is a motif involved, we think, in translocation of the protein, pathogen proteins from the pathogen into the host plant cell, is RXLR EER. And most of the resistances that we have in potato are recognising this kind of protein from the pathogen. They are in areas, and the font's probably too small, but that's 60 kb. So sometimes these genes are in areas where they're 60 kb away from their nearest gene neighbour. So it's very gene-sparse regions that are very repetitive, and that's why Phytophthora can shift them about. So what are the pathogen effectors and why are we interested in them? So pathogen effectors possess a multitude of effectors. So some bacteria only have 30. This kind of RXLR we found in Phytophthora, we found 500. So there's a lot there that we have to investigate. And what we think they're doing is they're going into the plant and they're manipulating the systems and the signalling within the plant for their benefit. And I'm just giving you some terminology. We're going to describe the ones that are not recognised as virulent. But the way the resistances work, this is just a bit of background, is that if we have a resistance gene, it's a gene-for-gene interaction, and that's the old reference that Angela will recognise. And that if we have a resistance gene there, it will recognise this effector protein, and you get a cell death, and that cell death stops biotrophic pathogens, pathogens that need living tissue to spread. This is a really effective way the plant has to stop them growing. We have this set of plants called the Black's differential set, and basically it's 11 different potato cultivars, each with a different resistance gene. And we can take Phytophthora that we collect from the field, spray them on this 11, and it tells us what effectors the pathogen has. So here, R8, this plant survived and the rest are pretty much dead. So this means that this Phytophthora has the effector AVR8 that's recognised by this resistance gene. And this has been the set of resistance genes that has been in most of the breeding material that I'm going to talk that's been used since the 60s. This is what the potato geneticists rely on. But these are the resistances that are breaking down in the field. Again, I'm going to give you a little bit about the life cycle, in that Phytophthora is growing in between cells, but they're poking feeding structures into cells at these points and at this early point here in the epidermis. These are the feeding structures, and these are the points we think these effector proteins are being delivered. And this is from a book chapter. I might have to press something here that we just recently wrote. So this is what happens in real time. We're actually expressing an effector and a resistance gene through bacteria here, but you can see you don't see anything when you're doing this for 24 hours. And then all of a sudden, the cell death is induced, so you'll start seeing it about now. And very quickly, the region of leaf tissue just suddenly dies. This is the defense mechanism that we're most interested in. But while we're doing that, I'm going to talk about the enemy's targets. So what we're interested in are core effectors. And what I mean is, I said before, Phytophthora has 500 RXLR effectors. They have other kinds of effectors as well, but we're interested in the RXLR effectors. And basically, we don't think all of these are expressed. We don't think every isolate of Phytophthora has all 500, and we don't think all of them are essential. So what we've been trying to do is get a core list by using microarray data, RNA sequencing, to try and find the ones we know are expressed in several isolates. So we also want to see the ones that are conserved. They're in the genome of as many isolates as we can sequence, or through markers. So there's people doing markers. We also want to see if they're essential. We use RNAi silencing techniques, both transient and stable transformation. We can transform our pathogen. And if we knock out one of these effectors, maybe the Phytophthora can't grow as well anymore. So when we've done these three things, we have a list still of 60 effectors that we want to investigate. And this is what our group, our institute's basically working their way through, looking at what these are doing. So we're using them as toolkit, basically, to try and work out what the Phytophthora wants to manipulate inside the plant. And we're doing this by, first of all, looking at their protein targets, so protein-protein interactions. There'll obviously be some effectors that target the DNA, maybe RNA as well, but I'm going to just talk about ones that attack other host proteins. So the first effector that I'm going to talk about has been published a while. This was in the lab when I started. AVR3A is one of the known ones. And here's the signal peptide that it's on the protein, and that's for secretion out of the pathogen. Then here's our XLR EER motif. And then the second half of the domain, the protein, is basically the effector domain, and that's the bit, the interesting bit, that's the bit we think is doing something. And it's recognised by R3A. We have 2 form alleles of this gene in all isolates that we've managed to sequence. We have a Ki form and an EM allele. And basically, I'll show you here with the lighting, hopefully you can see that Ki here is recognised by R3A, but the EM form completely evades recognition. So my message from that is 2 amino acid changes in this gene, in this protein, and basically it's not recognised anymore by the resistance gene. We went and looked at a number of isolates from Mexico, the source of Phytophthora, and you can see that most isolates contain the EM, the non-recognised form. And this is why resistances are breaking down in the field, because we're not understanding this kind of information before the resistances were deployed. Now looking back, you wouldn't put a resistance gene in the fields when you knew that the majority of the isolates out there had something that wasn't recognised. You're just asking for the selection to happen. So this is the sort of situation we're trying to avoid. Anyway, so back to looking at how this effector is working. We did protein-protein interaction screen using UCD hybrid. I'm hoping you guys know what that is. I haven't put a screen on how it works. But we pulled out 13 interactors, so it's not that straightforward what we think it's doing. And the one candidate we decided to characterise first was this one called CMPG1, and that's because it was a known protein involved in plant defence responses. What it is a E3 ligase, so for people that don't do molecular stuff, it's really protein turnover in the plant. Every protein that's made has a sort of lifespan of how long it's supposed to hang around, and E3 ligases are part of controlling how long a protein stays around for. So somehow this AVR3 effector is manipulating CMPG1 protein turnover within the plant. However, what we did find, which was surprising, is that AVR3 did not directly interact with the R gene, and none of these plant proteins interacted with the resistance gene either, so it wasn't quite as straightforward as we thought it was going to be. I did do a paper after that trying to characterise, try to find which cell deaths this protein was involved in. Here we have the major R genes of the potato, and they're mainly working in the cytoplasm of a plant cell, and it didn't seem to be involved in any of them. But what it is involved in is a lot of the early signalling, the sort of toll receptor-like, the PAMP, we call it. That is the recognition of sort of non-self molecules. So a plant can basically tell whether something's a bacteria or because of its flagella or things like that. So these early PAMP responses, that's what this CMPG1 was involved in. So perhaps AVR3A is trying to manipulate the early detection of itself, but it's got nothing to do with the resistance gene. So we were no further forward in trying to work out how this resistance gene is working. So we're still working on that. Also, the other surprise result that we got was when we knocked out this gene, our Phytophthora stopped growing better. So the dogma in our field at the moment is that host targets are there to be manipulated and probably to be knocked out, to be turned off, to be shut down. But what we found is if we knocked this out, the actual Phytophthora didn't grow as well. So what it's suggesting to us is that Phytophthora actually needs this plant protein to cause disease. It wants it there, and it's probably going to try and change it in a positive way. And I'll come back. That's one of the other messages from the other stuff that we've been doing as well. So here's another one, AVR2. So this is my baby. This is the thing I've been working on for most of the time. I'll only tell you a fraction of what we've been doing on this so far. But again, it's got 2 alleles, all isolates that we've sequenced so far. Obviously, that's not everything, but we can only find 2 forms. And instead of 2 amino acids difference this time, there's 13. So it's still not a big change. And most of the changes are in the C-terminus sequence. Again, it's quite black and white. The resistance gene R2 recognises one, and it doesn't recognise the other one. So we really see death, no death. And what does it interact with? That's what we wanted to know. So both the recognised form and the non-recognised form both interact with this one protein called BSL1. And this one is a phosphatase, and it's quite a unique phosphatase. There's only 4 of these in the Arabidopsis genome. They have this Kelch repeat at the beginning and this phosphatase domain at the end, and there's only 4 like this in the genome. We showed that this interaction happens in plants through pull downs, and basically we've examined the R2, the resistance protein R2, and the interaction with this BSL1 in the presence of the recognised and non-recognised form. And basically what we found was that BSL1, this target, only interacted with the resistance gene when the recognised form was there. Sorry, I've got a model, so maybe that'll help. So basically R2 is recognising something happening to BSL1 when AVR2 is present. This is what we think is happening. When we silence the BSL1 gene again, transiently, we can do that, we then lost the R gene recognition. That's what we expected to see for R3A, and we didn't, but this is what we're seeing here. If we silence this host protein, the R gene no longer works. So there's definitely the resistance gene is somehow monitoring something happening to this host protein, and we call this the guard hypothesis. So I put a wee slide for the non-molecular people. Sorry, this is from a lecture, so someone might recognise it. So we think the resistance genes are scanning the environment for changes, and what we thought is that they were directly recognising the effector. But now what we're thinking more and more is, if it's going to work, is that it's actually recognising a change to the protein that the effector is causing, a change to its own protein, not recognising the effector from the pathogen itself. And if anyone wants to read up on that kind of principle, that's referenced there. We've also used, we can overexpress our effectors, and we use bacteria. So again, you're using another pathogen to overexpress, so it can get a bit tricky. So on one side is our control, where we have a bacteria overexpressing an empty vector, and on the other side we have our effector of interest, and we've been doing that for all our core effectors. And basically we put Phytophthora on top of that, and you try and see if Phytophthora grows better when you've expressed that effector beforehand. And basically what we found in this case was that AVR2 and the host target, if we overexpress that as well before we put Phytophthora on, the Phytophthora grows a lot better. So again, it's the same result whether we overexpress the effector or the target. So it's another example of the target being needed to be there to be positively influenced by the effector. And again, that was not what we were expecting either from the dogma. So why would we think Phytophthora wants to increase the activity of this protein? And this is what we're trying to work out in the group. We're trying to do phosphatase assays and different things. This protein is involved in steroids, and for those of you who are not really plant hormone specialists, steroids basically promote growth. There's a long list of things that's involved in, but there's people here that are already know a lot about steroids, and what we think is it can both positively and negatively influence defence. So there is crosstalk. Obviously it's a pro-growth, and if your defence response is death, there's obviously going to be a conflict. And a lot of people are investigating the effects of steroids at the moment in plant defences, and this is some of the things that we're doing. This is the pathway. It's a phosphorylation cascade, so I'll not go into too much detail, but our BSL1 sits right there in the middle of a phosphorylation cascade, downstream of a receptor called BRY1. This is my last one. So we've got so many effectors, we stopped giving them names, so they're just numbers now. So most of the papers you'll see coming out of the lab in the last 2 years will all be just numbers, but this is the genome ID number, so it does help you find it. If you ever want to go to the Phytophthora genome database, you can plug that number in and you'll know exactly what protein I'm talking about. Here, this one is interacting with a protein called NPH3, so this one's just in press, so it should be out. And it's to do with blue light signalling. Again, we're not really sure why blue light is being targeted, so this is a current activity in the lab, as we'll be looking at light. This is the yeast to hybrid again. You see it going blue when there's an interaction. And we also use split YFP quite a lot, and this can tell us where the proteins are interacting. So these kind of pictures never usually show up that well with lighting, but you can see it's in the cytoplasm. This is a close-up of the nucleus, and you can see that it's round the nucleus, not inside it, so we can tell that when the effector and this host protein are not going into the nucleus together. But, so I just put this in for background if anyone's interested. So this is the MPH3. It's phosphorylated at night and it's dephosphorylated during the day. Again, there's part of the E3 ligase activity we think it's having. So it's another E3 ligase protein turnover, but to do with light sensing. So I was going to spend the rest of the talk, as I gave my title, talking about these three things in more detail. But I thought, since also there's a lot of more applied research here too, I thought I would go into more detail about our strategies and how we're going to use it for trying to plan more durable resistance. So the take-home message is that we are finding susceptibility factors. We thought effectors would target proteins to knock out functions in the plant, but it looks like a lot of the things that we're finding are actually required for the disease to happen. Pathogen wants them there, and they maybe want to activate them or change what they do, but they need them there for activity. And you can read that. That's another review we've just written, which should be out now. So, resistances. So potato, you were talking about potato. We are big fans of potato and we love eating it, but it's really beginning to increase production in the developing world. So India, China are really taking more interest in potato. You can get all sorts of crazy products. Here's a potato drink that they like. You can buy in a can. There's potato hot drinks. You get these potato flakes and they make pink and purple dumplings and bread. So they're using potatoes for a lot more than just chips and crisps and boiled and mashed like we do. But they're not quite as good for getting their yields up yet in the developing countries, and we are helping them with that. We have collaborations, and they're also very interested in reducing their inputs, increasing their resistance to biotic and abiotic stress. So the same aims and goals as we have. Again, the potato genome sequence has helped us with this. Again, it was big, so it wasn't sequenced till quite fairly recently, and it's a lot bigger than the model plant Arabidopsis again, which is why it was a wee bit later coming out. At our institute, we have a lot of potato geneticists. There's about 54, 55 people working on potatoes at the James Hutton Institute, and they're working on defences, like I talked before, and the genes that we're interested in are spread across lots of different chromosomes. There's also people looking at shape, skin colour, drought, heat, cold stresses, and nutritional qualities. And sometimes we all meet together and try and write grants that cross these themes. And I'm beginning to talk a lot to people working on drought, heat, and now light because of the light sensing that we're finding. Here is what a resistance gene looks like in the domains. Here's the signalling domains, the activation domain. This is needing a nucleotide binding site for energy, and the recognition part. And this is the bit that we think recognises changes in the host proteins. Again, like I said before, there are people at our institute working on bacteria, viruses. So it's not just Phytophthora at our institute. There's also people interested in the resistances of the other genes as well. So how many resistance genes do we think are in potato? At the moment, we think there's about, well, there's different opinions, but there's about, say, 500. And they cluster in this sort of family tree. So here's our two I talked about earlier. The vector that I work on is recognised by this cluster here, and that's the biggest cluster here. I also talked about AVR3A, so that's the R genes that recognise R3A. So there isn't just one there. There's sort of like a family of sequences, and the struggle is knowing which one's the active one. And Angela will know the trauma you can have with doing that. At our institute, we have a Commonwealth Potato Collection, CPC is for short. And basically, we had scientists go off to Mexico and South America and gathering wild potato accessions and early cultivated land races. So we have about 1,500, and we grow a small proportion of that every year to keep the seed going. And of course, being interested in defence, we want to see what defences resistance genes are in this collection, but we can't go sequencing every single one of these 1,500 accessions because we'll be there, well, we'd be in a job anyway for the rest of our lives, but it costs too much money. So what our department are trying to do, actually my husband is trying to do, is find ways of sequencing the bit of the genome that we're really interested in and get the most information we can without having all the rest of the bioinformatics to worry about. Only about 20.24% of the potato genome is actually resistance genes. The rest of it we don't care about. So 99.75% of it would go in the bin. And there's a lot of money to spend sequencing a genome if you're not interested in it. So what they've used is this Agilent Sure Select system is this techniques we've used. And basically they're RNA probes that are biotinylated. And you have to design them. There's a design phase. If you've got a family of proteins that genes you're interested in, you have to sort of design probes with them. So this is 48,000 probes, and they're only 120 nucleotides, and they will recognise they're designed to the resistance genes that we're interested in. And basically, you shear the DNA, you incubate it, and you pull out with streptavidin, and you only sequence the stuff that comes out with your RNA probe. So you're only sequencing the genomic DNA that you're interested in. So here's an example of the potato genome before we reannotated it. And here these peaks are read depth. So that's the other good thing, is we're getting good read depth from this. You can see where our genes are. And then when we did this on our DM variety, this is double monoploid potato, the sequenced one. Ingo, my husband, managed to reannotate several new R genes in places that nobody had identified. Also in gene sparse regions, these are areas where no R genes, resistance genes were mentioned at all. You can see we've got reads coming back from our probes. And then, so we identified more R genes in regions where there weren't even any R genes mentioned. So if you go to this paper here, you'll see that and here shows as well that the green ones, they were previously annotated in the potato genome paper. And by using these probes, we've managed to almost double the number of resistance genes that we know are in this genome. And they're shown in purple. So hopefully everyone can see purple and green. So we've almost doubled the number of potatoes. And 50% of all the reads we got back from this were our NBLRs, so only 50% went in the bin compared to 99.75. So we find this really a bioinformatic challenge. So to do this kind of research, you really need good bioinformatics. You need a good genome sequence, and then you can make progress. And we're still really only getting it to work well in diploids. Tetraploids or others might be more difficult. So it's a bioinformatic challenge rather than a biological lab challenge at the moment. But here's a sort of diagnostic way we're using this as a diagnostic tool that I wanted to mention to you. So our department have this Solanum ocadi. This is a wild species. We know that it has several accessions in this species, and we know one of them is resistant and has the resistance gene that recognises AVR-VNT1. If I bring this up, we have some other accessions. We know they recognise AVR-VNT1, but we don't know what's in there. So is it worth our time spending a lot of time breeding with this material when we don't know what resistance gene is inside it? So basically we've used these probes with known resistance genes, and we know they cover R1, 2, 3, as I talked about before. And our resistant material has this full length. You can see we have reads covering the whole genome sequence of VNT1. And here's our susceptible accession, and it's missing a fragment of the gene at the 5' end, so it's truncated. When we looked at our resistant and susceptible bulks from the progeny, we see that all the ones that are resistant have a full copy, and all the ones that were susceptible also had this truncation. So also there was a PhD student in the lab, and she was trying to clone manually with traditional techniques this VNT1 from these different accessions, resistant accessions, where we didn't know whether VNT1 was there. This took her 4 months to clone VNT1 gene from there and be able to say, yes, it was there. This took the bioinformatician 4 hours and with her software. So if you've got a DNA sequence, it really does speed up what we can do. So again, just a take-home message is that we're using plant resistance accessions. We're looking for our genes, but coming back to what I do, we're using the pathogen effector sets that are expressed essential, and then we're looking for resistance genes that recognise those core set and feeding back. So the potato geneticists and the pathologists are working really nicely together at the moment, trying to speed up how we're going to identify and deploy these techniques. So we have a pipeline really, and it does involve genome sequences and bioinformaticians, but we still need the biologists in the lab to characterise things, so there's a lot still to know. So here are my main points, is that oomycetes are very devastating diseases of many organisms, not just plants. Late blight's one of the most historically significant one. We're understanding how the pathogen effector research can tell us what kinds of pathways the pathogen wants to manipulate within the plant. We're wanting to find effectors that recognize, resistances that recognize these core effectors. We're looking for smarter ways to sequence genes and to speed up deployment in the field. And I'm going to do my plug for GM. I still think that we better address some of these traits through biotechnology solutions. You can't breed a resistant variety and expect it to still work 25 years later when the pathogen's changing in the field all the time. It needs to be more like the flu vaccine. You know, when people know the particular flu virus is circulating the world, within 6 months, a year, they've got a vaccine for it. That is almost, we need to do that in agriculture as well to really beat these pathogens without chemicals, I think. So, or even pyramid resistances as well. So this is the team of people that really work on the plant effectors and what they're doing in the lab, but we're a big group of people that are studying the Phytophthora, the populations in the fields, the resistances, markers, genetic facilities, microarray facilities. So I can't list everybody, but there's a lot of people that are involved in the work. So that's it, and if anyone's got any questions.